For most of the semester, we talk about the prokaryotic microbes. More specifically, we actually talk just about the bacteria, not so much the archaea. However, as we learn in Chapter 1, there are other groups of microbes out there, and in Module 7, we talk about the eukaryotic microbes and the viruses, and some of the things that they do, and then eventually we might refer back to them throughout the semester, but for the most part, we go back to the prokaryotic microbes, again, our bacteria. So we'll go through two chapters in this module, chapter 12, chapter 13, and we're just going to distill it down to just the basics. There's a whole laundry list of eukaryotic microbes, and there's a whole gigantic way of talking about how viruses divide, but we're not going to talk about the nitty-gritty details, just a general survey so you get an understanding of what some of the eukaryotic microbes are, how they differ from the prokaryotic microbes, as well as viruses. Some of the things they do, some of the generalities, and then so then we can get a general understanding of some of the other microbes. So we'll start with fungi. And recall from chapter 1, mycology is the study of fungi, right? So myco is the root for fungi, ology, of course, is the study of. And fungi are very important in things like the food chain. They're very important in decomposing dead matter, recycling vital elements. But they're also used for food, and of course we know mushrooms are fungi. A lot of people eat various types of mushrooms. Some cheeses have fungi in them. They help us produce food, so things like bread. Yeast is a fungus, right? So anything that yeast makes are produced by fungi. And a lot of our antibiotics, right? So penicillin, for example, initially comes from a fungus called Penicillium. But it also we also use our fungi to produce antibiotics for us. Right? So many of our antibiotics are produced within fungi. Fungi are chemoheterotrophic, which means, of course, that they eat chemicals and they are organic, right? Very much like us. And for the most part, because of the fact that they're chemoheterotrophic, they compete with bacteria for food, right? So they don't have their own little metabolic niche, so to speak. They have to compete. And later we'll learn about some of the things that they can do in order to compete. Again, very quick list of fungi versus our bacteria. Remember, fungi, eukaryotic, right? Chitin cell walls. They do not have peptidoglycan. Because they're eukaryotic, they have sterols within the membrane, which bacteria do not. And they can produce not just asexually, but also sexually. Right, so they're able to reproduce sexually, which is a new thing for us, something we haven't really discussed yet. And we'll later get to talk about what it is or the purpose of sexual reproduction as opposed to just asexual reproduction. Fungi, many different ways to classify fungi, but we're going to talk about one major way, and then that's all based on what we call multicellular idea, or what do they look like on a macroscopic level? What do you see with your eyes? And in this case, we can define them or classify them based on how big they are, what do they look like when they're growing, and what type of spores do they produce. So we can classify them into three groups: the molds and fleshy fungi, the yeasts, and the dimorphic. And dimorphic essentially means they can do both. Right? So we're looking at molds, which are multicellular. Oops, I spelled that wrong. Multicellular. Maybe I'll just rewrite that. Looking at molds and fleshy fungi, which are multicellular, or yeasts, which are unicellular or single cell. And that's kind of our differentiation line. And again, dimorphic will do both. So let's start off talking about our molds. And our molds, when we refer to the organism or the body of the fungus, we call it the thallus. Right? So the thallus is the body of the organism. And the thallus for molds and fleshy fungi consists of these long filaments called hyphae, or hypha for singular, right? So many hyphae, one hypha. And typically what will happen is that the fungus will grow out from a center, form these filaments, very much like that. And these mass growths are what you see growing on your food or in your bathroom tiles or wherever you see them. And then this whole mass together would be called a thallus. Now these of course will split off and start a new thallus elsewhere. And usually what you end up getting now is once it becomes visible, you don't see one single thallus, but many together, forming this one mass filamentous mass of fungi. And we call that the mycelium. Right? So here what we're looking at is a mycelium. Whole bunch of fungi all stuck together, a bunch of thalluses. And then within them are these individual strands of hyphae, both vegetative, which means actively growing, and aerial, which is used for reproduction. But we'll get to that in a second. Hyphae grow by elongating at the tips. Right? So each tip simply just grows. So if we have, let's say, a hyphae, which is actually composed of cells, a hyphae simply grow by elongating at the tip. And each tip of the hypha is capable of growth. So you can grow this way, or you can grow that way. And this is what makes it so difficult to get rid of fungi, molds in your bathroom, because let's say you're scrubbing and you're cleaning, and then you get rid of all the fungus except for one or two cells. Well, these one or two cells can continue to grow, right? And they will continue to grow until you're able to get rid of them all. Again, that's what makes it so hard to get rid of. The term vegetative means actively growing, right? So vegetative hypha is the part of the hypha that's actively growing, the part that's obtaining nutrients. Now there's two types of vegetative hyphae. Most are called septate hypha, or septate hyphae for plural. Right? And that's because they have these things called a septum, septa for plural. So this septum separates the hypha into these cell-like units, right? And most of these have one or two nuclei, whereas cenocytic hypha are not as common, and cenocytic hypha do not have any septa, or no septum in between the cell lines, right? So they look as one gigantic mass. They do, of course, have cells, but it looks like one gigantic mass with many, many nuclei. Right? So these are two types of vegetative hyphae. Again, the part that's elongating, and like we've discussed, growing out at the tips, right? So that's how they'll grow. Elongating, just looking to get bigger while they're feeding. Reproductive hyphae are often called aerial hyphae, and that's because aerial hyphae project off the surface. And this is a nice advantage for fungus, right? Because they'll be able to come up and off, and then once the spores or the reproductive structures are out at the end of the thallus, or at the end of the aerial hyphae, it makes it much easier to spread. When they fall, even something like that, that's a great distance for a single cell. Or if, let's say, an insect is walking by and the spores that are higher up will get picked up on the insect, they'll walk very far, and then they can deposit those spores elsewhere, which then can grow into a new body or a new thallus. Right? So the fact that it's able to come up off the ground, off the surface, is a great advantage for fungi and their ability to not just reproduce, but to spread distances. And that's ultimately what you want to do. You want to spread and see the world, right? And the aerial hyphae are typically what you see. So next time you have some mold, just take a look at it. You'll see these little things sticking up off your bread, and those are the aerial hyphae sticking up and off. Yeasts are unicellular, or single-celled. Right? So yeasts, unlike our molds, the whole thallus consists of just one cell, right? So uni or single cell, which then of course means that they're non-filamentous, right? And these are microscopic, so in many ways they grow kind of like bacteria. We can't really see them. We grow them in the lab just like we do with bacteria because of the fact that the one cell divides. Yeast often divide by this thing called budding, where the bud just kind of pops off, and it's not quite like binary fission, where one cell splits evenly into two. With budding, this little thing comes out, and then the daughter cell is often smaller than the parent cell. Right? So it's not quite even. But again, many ways the rate of budding is consistent or constant, and therefore we can kind of make predictions about generation times and things like that, just like we do with bacteria. Most yeasts are capable of facultative anaerobic growth. Right? That means that they can use oxygen as a final electron acceptor to produce carbon dioxide and water. And here I've left the question, what type of metabolism? So I'll let you think about that for a second. They can also use organic compounds as a final electron acceptor with products of ethanol and carbon dioxide. And again, I've left the leading question here. What type of metabolism? Now, for the most part, we prefer this type of metabolism, or number 2 type of metabolism, for industrial purposes, because it's the more useful of the two, because of its products, ethanol and carbon dioxide. So I've left you to kind of think about the two. Have you thought about it? You can pause it if you like. This is aerobic respiration. And this here is fermentation. Right, or if you want to be more specific, ethanol or alcohol fermentation. And it's our ethanol or alcohol fermentation that leads to things like beer and wine production and breads and various other things. Again, very important in the food industry because of the fact that it's this ethanol fermentation from yeast that ends up producing much of our food products. Dimorphic fungi grow both as yeasts or molds, right? So they can grow as single-celled organisms or as multicellular filamentous organisms. Pathogens, often dimorphism is temperature dependent. So in the body, when conditions are great, lots of food, it's always nice and warm, tend to grow yeast-like because they are less dependent on others. They're very capable of growing by themselves. But when they're outside the body, where resources may be scarce, they tend to grow more mold-like, right? And that's in that environment now, it's easier or better to survive as a whole. Right, so these are a little bit more difficult to identify because of the fact that they can do both types of growth. Fungi, again, are chemoheterotrophs, and that means that they have to compete with bacteria for food, which if they were simply to go head-to-head, you say you have a fungus here, a bacteria here, and then you say, go. Bacteria would win out because they're able to grow a lot faster than fungi. And therefore, they would eventually swarm over the fungus, take all the food, and win out. So the fungus needs some sort of competitive advantage. And in the fact is that fungi have these nutritional characteristics that does allow them or provide them some advantages. So for example, fungi can grow at pH 5. They can tolerate more acidic conditions, whereas most bacteria, they don't really like it at that level. They grow much too slow at pH 5. Fungi are very resistant to osmotic pressure, right? So they're more resistant to plasmolysis. And bacteria are less tolerant of these types of things. Fungi in general are psychotrophic, right? We should look up what that means. But that means they can grow at moderate temperatures. Room temperature is perfect for them. They love that. So they can compete in areas where outdoor it's not going to be quite as warm. Fungi can get away with areas that are pretty low in moisture, low in nutrients, and that's because they grow slowly. Right? Bacteria, one of the disadvantages of growing so fast is that if the conditions are not right, eventually you just sort of outgrow your ability to grow. Right? Whereas fungi grow nice and slowly, right? And because of that fact, they don't. They can kind of take their time. Right? So if there's not a lot of nutrients, not a lot of moisture, then they're okay with it. And that's one of the reasons why, by the way, they can grow on things like your bathroom tiles. So you ever wonder, what is this mold eating when it's sitting here on my bathroom tile? Well, pretty much it's just sitting there, waiting for something to come along. And then when you're showering, and then the skin cells come off and then lands on the bathroom tiles, well, then they get to eat a little bit, and then they kind of sit there and wait until more comes along, right? But they take their time. Here's a nice little experiment that you can do. This is Jell-O. Jell-O is a very interesting piece of food, right? We enjoy it. But Jell-O, even though it's rich in sugars, is actually not that hospitable of an environment because of the fact that there's so much sugar. Lots of solutes, right? When there's lots of solutes, what does that mean? Hypertonic environments, right? So overall, bacteria don't really spoil things like Jell-O very well. Or at least it takes a long time. Right? And the first things that end up spoiling your Jell-O, you guessed it, the molds, right? Because they're more tolerant of things like osmotic pressures. Right? So they're capable of growing in this. This is a little Jell-O that I made. I added some fruits, increases the acidity. Right? So this is slightly acidic, high in osmotic pressure. And I left it in the fridge. I left it in the back of the fridge for a long time. And invariably the first things that go on there, always things like this, right? So these are molds. This guy here is actually Penicillium, and he's the guy that probably spoils most of our things first. gets in just about all our foods that we eat. And so next time you make some Jello, just take a small little cup, put it to the side, and see what happens. Right? These are little everyday science experiments that we can do. All right, let's talk about the life cycle of the fungus. And for fungi, reproduction is all about spores, right? Not endospores. Endospores are those internal resistant structures. These are external structures used to reproduce, right? And essentially what we do is we form these spores. These things then eventually detach, land somewhere, and then they germinate to form a new fungus. Just like that. Now, teleomorphic fungi can do both sexual and asexual reproduction, whereas anamorphic can just do asexual reproduction. Now, at this point, I want to talk about why do both? What's the purpose of asexual? What's the purpose of sexual reproduction? And for everything, especially in biology, there are always advantages and disadvantages. Everything is always in shades of gray. Why would you want to do one? Why the other? Why not both? So let's talk about sexual reproduction. Right, so we as humans, we do sexual reproduction. Right? We need two people to produce an offspring. And the main advantage of sexual reproduction is this key here. And it doesn't seem like an important advantage because we don't really see it. But genetic diversity is hugely important for survival of species. So when a species grows, the more diverse your gene pool, the more capable they are of resisting harsh environments, harsh stresses that may affect a certain species. And so sexual reproduction really is good at increasing genetic diversity. And for microbes, it's hugely important. For malaria, for example, malaria is very hard to treat because of the fact that it undergoes asexual reproduction. And we'll talk about malaria a little bit later. Did I say asexual? Malaria, or Plasmodium, is capable of undergoing sexual reproduction, which then increases its genetic diversity and making it very difficult to find drugs that we can use to eradicate malaria. There is a major problem with sexual reproduction. And what do you think the problem is with sexual reproduction? Well, it's got something to do with the fact that it's hard to find a mate sometimes, right? So it's much easier to do asexual reproduction. So asexual reproduction is easy. Purpose of asexual reproduction is to increase population size. And asexual reproduction is fast and easy. You just pretty much divide whenever you're ready. And it's a great way to increasing population numbers. And actually, so even though us humans as a species produce sexually, if we want to produce our offspring, that's a sexual reproduction. When we grow, much of our growth is actually asexual. We have these cells that simply divide. Right? So we don't need to constantly be fusing two cells in order for us to grow larger. Right? When we want to make more skin, make more hair, our cells simply split, and that's asexual reproduction. Great way to increase numbers. Right? So for organisms that can do both sexual and asexual reproduction, it's a great advantage because you can have a part of your life cycle where you simply increase your population size, and then you can have a dedicated part where you can now try to mix some genes, get some genetic diversity happening. Right? So if you find a mate, do some sexual reproduction. If you don't have a mate, then you do some asexual reproduction. Let's talk about asexual spores first. So asexual spores essentially form by fragmenting hyphae. Right? So we have our hyphae that are growing. And then when it's time, we fragment them. Right? So we form this hard shell around the hyphae, and then they can kind of break off. And there we go. We got our asexual spore. And the most important thing about this is that spores are genetically identical. Sorry, asexual spores are genetically identical. Again, the idea is that we'd like to increase population size, but very quickly. But the cost of that, of course, is that it doesn't increase our genetic diversity, right? So we have this increase in numbers, but all the spores are genetically identical. Now, the first type of asexual spore we'll talk about are conidiospores, and conidiospores are spores that are not enclosed in a sac. Right? The simplest kind of spore, basically we have these conidia or conidiospores that are produced at the end of this thing called a conidiophore. So this conidiophore is our aerial hyphae, and then you can see how we have this hyphae growing here with these spores at the end, right? So these spores have fragmented and are just kind of at the end, right? So these are naked spores. These things are ready to disseminate. Arthrospores are related to as a type of conidiospore, and rather than now forming these, rather than forming these chains of spores, arthrospores form these large thickened spore. We call this an arthroconidium or an arthrospore. Finally, our last type of asexual spore we're going to talk about is called the sporangiospore. And sporangiospores are formed within a sac. Right? So within this large sac called a sporangium, at the end of its sporangiophore. Right? So again, this is its aerial hyphae, the sporangiophore. Form this sporangium or a sac, and inside are hundreds of sporangiospores. Right? So these spores are, you can see them through the sac, but they're all on the inside of this sporangium. And so that's kind of your basic type of asexual spores, enclosed within a sac or not enclosed. And there are lots of other types of asexual spores. We'll just kind of leave it at these three. We got our naked spores in a chain. We got our naked spores, one really thick, gigantic spore, and then those that are enclosed within a sac. We'll talk about some sexual spores now. Right? So again, sexual reproduction is all about mating, right? So we have two parents, plus and minus, and spores have characteristics of both parents, right? So every spore, and this would be a sexual spore, is different. So every spore is different, but the drawback is that you need to find mating types. You need to find a minus if you're a plus, or a plus if you are minus. Let's see, let's talk about a couple. Zygospores are simply fusions of haploid cells, which means single copy genomes. Right? Basically, we have these two cells that come together. Oops, how did that happen? Plus side, minus side, or vice versa. And then they fuse together to form this zygospore in the middle. Right? The zygosporangium, which contains the zygospore. And again, this zygospore will have characteristics of both this and this fungus. An ascospore is a sexual spore that's contained within a sac called an ascus. Right? So here we have an ascus, the sac, and these sexual spores are contained within. Right? So these ascospores are contained within a sac. And finally, we'll talk about a basidiospore. Basidiospores are formed on a pedestal, and this pedestal, this basidium, is what we call a mushroom. Right? So within this mushroom, usually sometimes underneath, sometimes on top, are your basidiospores. And again, this projection off the surface allows the spores to be disseminated. And many mushrooms are edible. And the reason or the purpose for them being edible is that they can be eaten by birds, and the birds will take them away and then end up dropping them great distances elsewhere. All right, we'll end our section on fungi, on fungal diseases, because ultimately we want to know how do these things affect us, right? And a fungal disease is called a mycosis, or mycoses for plural. And generally, fungal diseases tend to be chronic or long-lasting because, like we mentioned earlier, fungi tend to grow slow. And many of our antibiotics, if you recall, are dependent on the fact that things are growing. Right? So if things aren't growing, it's not going to work. So we can classify mycoses basically into 5 groups, all according to how much of the tissues are involved and how does the fungus get into the host, right? And they're kind of, I think we're going to go more or less from order of most severe to the least severe. So we'll start with a systemic mycoses, or systemic mycosis. And a systemic, I like to call it system-wide. And if you know what system-wide refers to, basically it infects the whole body, right? So systemic mycosis is one that infects the whole body. And the predominant entry is through inhalation. So often the fungal spores will start in the lungs and then create either some sort of neurotoxin, or the spores will spread to other tissues after getting into the lungs. And so systemic mycoses are the most severe, something that affects the whole body, many, many tissues, very dangerous type of thing. Subcutaneous mycoses are ones that are beneath the skin. Right? So the cutaneous layer refers to the skin layer. So every time you see the word cutaneous, that means skin. And sub means under. Right? So these are now infections under the skin. And these are essentially via punctures. Right? So the punctures are then able to get the spores underneath the skin and then cause infection down there. Right? So under the skin. Now it sounds similar to, but it's different from a cutaneous mycosis, or sometimes called a dermatomycosis. And again, cutaneous referring to skin, right? So now this is an infection of the skin, and that's because here now the fungi are eating proteins, often keratin, that are found in your skin and also your hair and your fingernails, right? So now this is an infection of the epidermis, which is your skin layer, and if you pinch your skin, it's that part that you can pinch, as well as the hair or your fingernails, because that's where keratin is found, and it's transmitted by contact. And that's often just getting onto your skin and then ultimately eating, again, your skin. Again, this sounds similar to our next one called a superficial mycosis. And in a superficial mycosis, we're talking about on the surface, right? If you're a superficial person, you only care about what's on the outside of the person. Superficial mycosis is just about on the surface, eating the dead skin cells, eating the stuff that's just right on the outside. Right? So this is now localized on the hair shafts, not the hair, just the hair shafts. Right. So if you have, let's say, your skin, and then we got our hair shaft, just growing on the inside like that, not really caring about this hair that's grown out of it, or just sitting right on top of the surface of the skin cells. And things like dandruff is a superficial mycosis, or it can be in part. Just sitting there eating the dead skin that's on top and then causing this flaking. And sometimes, because it's so harmless, people are often unaware that they even have a fungal infection. You think, oh, I'm just flaking a little bit, and you don't realize that you have a fungal infection. An opportunistic pathogen is it's not so much an invasion of tissue here now. We're basically talking about fungus that's normally harmless, but then becomes pathogenic when a host is compromised or in a weakened state. So antibiotic treatment, for example, can compromise a host, or those with a suppressed immune system. Right? So, for example, pneumocystis is a fungus that's pretty rare. I mean, it doesn't cause too many problems for normal people. But for for AIDS patients who have a very severely suppressed immune system, pneumocystis can cause life-threatening infections. Stachybotrys grows in water-damaged homes. Again, doesn't cause too many problems, but for infants, it can cause some major issues because infants don't have a very strong immune system. Right? So very poorly developed immune system, therefore, can cause a lot of problems. Right? So 5 different types of mycoses. Again, kind of pay attention to how much of the tissue is being invaded, and how does it get into the host. I actually did say we were going to end the fungal section on the mycoses, but we're going to end it on some chemotherapy. Right? Just again, talk about some medicines, some drugs that we can use to target fungi. And because fungi are eukaryotic, that means their structures and their metabolism is the same as us, right? So it makes it very difficult to find chemicals that show selective toxicity. However, it turns out that the sterols that they use in the membranes are different. They use ergosterol, where we use cholesterol. So that makes a nice target, right? So many of our fungal drugs are ergosterol synthesis inhibitors. And of course, fungi have cell walls, right? The chitin cell wall. They're also a nice target because of the fact that we don't have chitin cell walls. Ergosterol and synthesis inhibitors cause excessive permeability of membranes because of the fact that we're removing the sterols. And again, very quick recap of what sterols do. Right? So we have membranes, our lipid bilayer, And I'm just going to draw the sterol as a box because I'm too lazy to draw the rings. But they insert into the membrane just like that. And this insertion of the sterols provides extra strength to the membranes. And if we can find ways to, let's change colors, if we can find ways to eliminate the sterols, all of a sudden now we have a membrane that's compromised. It's not as strong, not as protective as normal. And therefore, we can call these ergosterol synthesis inhibitors cause an increased permeability of membranes. Basically, creates a membrane that's not very stable. And the most common type used to be the polyenes. And the polyenes are a group of ergosterol synthesis inhibitors, of which amphotericin B is the most common. But it's fairly toxic to the kidneys, so we don't use it all that often, right? So it's good in low doses, but because, again, we need to often use these treatments for a long period of time, it's not the most, it kind of fell out of favor to what we call the azoles. And the azoles are the most widely used antifungals. Imidazoles are used in topicals, so anything that you buy in the stores for athlete's foot and yeast infections have imidazoles in them. And triazoles have very low toxicity, right? So it's right now it's the most commonly used antifungal medication. Oops. Cell wall synthesis inhibitors is all about inhibiting this molecule called beta-glucan. And this beta-glucan is found in the chitin cell walls. And again, incomplete cell walls can lead to lysis because we have an unstable cell. And these drugs, the echinocandins, they're useful for systemic mycoses, right? So because for systemic mycoses, which go all over, we need to make sure that we have molecules that can transport and take internally, right? So the echinocandins are often used for systemic mycoses. And again, what they do is they attack the chitin cell wall, prevent synthesis of it, right? And again, so this will only really affect this fungus while it's growing. All right, so that's the fungus, the fungi. Just related to that are the lichens. And a lichen are these really neat organisms. If you don't know what it is, you will learn. And if you do know what it is, you will know that they are everywhere, right? And we'll look at some pictures later. I have one picture. And you'll recognize, hopefully, almost right away. Lichen are a mutualistic combination. A mutualistic means, and we'll define this a little bit later in maybe our next module or the module after that. Benefits both organisms, right? So we have an algae. An alga is single algae. Or sometimes it's a cyanobacterium, which is technically a bacteria. Not technically, it is a bacteria. But we call it a green algae because this bacteria has chlorophyll, grows, or there's plant-like photosynthesis, right? So it pretty much is like this single-celled plant, like algae, and a fungus. And this mutualistic relationship benefits both. The alga provides carbohydrates that it gets via photosynthesis. Remember, it can make food from nothing. All it needs is some sunlight, and bang, it's got some food, right? Whereas the fungus provides what we call the holdfast. It allows it to stick to areas, make sure that the algae is always in where it wants to be to get the best photosynthesis going on. It also provides protection, and nothing gets swept away. It hangs on tight to whatever environment it's on. And this mutualistic relationship allows it to survive in areas that are actually kind of harsh, right? We're talking about rocks. And sometimes if you see these little crusty things on rocks, those are lichens. Or on the barks of trees, which are actually not very pleasant to live on because bark is not easy to digest. They grow there, and it's basically the algae there can't grow alone because it won't be protected. The fungus there can't grow alone because there's nothing to eat. But this mutualistic relationship allows both of them to survive. And lichens are actually very important in the north, in the way north, where there's no trees, there's nothing to eat up there. Many of the animals sit there and eat the lichens, which flourish there because lots of time to grow, right? Nothing there to compete against. Oops, here's what a lichen looks like. Right? These green cells are our algae, and they're protected by the fungus, right? The cortex of the fungus or of the lichen provides protection. And then down here at the bottom we have our holdfast. This is where they would stick. Let's see, 3 types of lichens. And all 3 are pictured right over here. And let's take a look at it. If you look at this picture, I bet you think I took this right here outside of my own yard. I don't have a yard, but I have trees in front of my house, right? And if you look at a tree, and if you don't do it today, do it tomorrow, go look at a tree and notice these things. They're growing everywhere. And they, again, growing and flourishing in an environment that's normally difficult to grow in. 3 types of lichen. I didn't label it because we can go through it together. Crustose lichen grow flat or encrusting on surfaces. Foliose lichen are leafy, and fruticose lichen have these branched projections. These projections that kind of stick out. Right. So let's try to identify them. Where is the crustose lichen on this tree? Boom, crustose lichen, right? Grows flat or encrusting flat on the surface, right? Crustose, crusty, grows on the surface. Foliose are leafy. Where is the foliose lichen? Right there, looks kind of like a leaf stuck on it. All right, and where's the fruticose lichen? The fruticose lichen are these things, these branched projections. And from what I remember, we see a lot of the foliose and a lot of the fruticose lichen on the trees. Right? So again, go find a tree, walk outside right now, look at a tree, and then you'll see these guys hanging out there. Notice the color of these. They are, in the case of the crustose, very green. In the case of the foliose and fruticose, greenish. Right? And the greenish pigmentation, or the straight up green, are the algal cells that you see. Alrighty, let's talk about some protozoa. And protozoa are very interesting. They're unicellular eukaryotes, or what we call our animal-like eukaryotes, right? The life cycle of protozoans can be quite complex, especially the parasitic ones. And the ones that do cause disease often involve so many different life cycles, multiple hosts, very complex life cycle. They all have a trophozoite stage, that's the feeding and growing stage, the vegetative part of the life cycle, and then often undergo many, many different changes of form. Most are harmless, just kind of doing what they do. Algae technically are protozoa, but we kind of treat them as separate. But the ones that do cause disease cause some pretty hardcore disease, right? Pretty significant ones. So we're going to talk about some of those. Let's see, our protozoa can reproduce both asexually and sexually. And again, what's the purpose of asexual reproduction? If you said to quickly increase population size, that is correct. And the purpose of sexual reproduction, if you said to increase genetic diversity, that is correct. Many protozoa can undergo an encystment stage, and the encystment stage is formation of this protective capsule called a cyst. Sometimes it's called an oocyst. It all depends on exactly what part of the life cycle comes in. But basically it's like an endospore. It protects the organism like a seed, and it allows the parasite to survive outside of the host where food is lacking, moisture is lacking, temperature, it's cold, poor oxygen, and that kind of stuff. And sometimes it's a very important part of its life cycle. Here we got Giardia lamblia, right? On the left, that's its trophozoite stage, actively growing. On the right, we have its cyst stage, right? And the cyst now allows it to survive, again, when it's outside of the host. And the cyst can sit there dormant. Giardia, by the way, interesting disease. We call it beaver fever. Back home, sometimes call it hiker's diarrhea, right? And it's commonly you commonly catch Giardia infection when you are drinking water out in nature. Right? You know, you're hiking along, you see a stream. It's like, oh, this stream is nice and clear, and because we're outside, it must be clean, right? So you drink it. Unbeknownst to you, of course, there's a pig or a mongoose upstream going doo-doo in the water. and then you end up sucking up the cysts, right? And the cysts allow it to survive in the water, which is super cold, right? There's not that much food, always moving. So you're drinking up the cysts, and then it sits there and grows your intestines and gives you some really bad diarrhea. I heard it's really nasty stuff. A friend of mine caught it once. Just really bad kind of stuff, right? So again, this idea that the cyst allows it to survive in areas where normally it wouldn't be able to. Let's see, we got protozoa are mostly aerobic heterotrophs. Some grow anaerobically, but not the ones that we're going to talk about. And all need to live in an area with a large supply of water, right? Because they don't have cell walls, they don't really have a way to protect themselves, so they need that moisture to survive, whether that's inside a person, whether that's some area that allows them to survive, or some area with a lot of moisture. Now, rather than talking about various life cycles and that kind of stuff, I thought it'd be it's more efficient maybe to talk about some protozoan diseases. And then looking at the protozoan diseases, we can get a good example of what is it that they do? How do they cause disease? So we'll start off with malaria, probably the most significant protozoan disease worldwide, kills millions of people get infected every year, hundreds of thousands of people die. And Plasmodium lives in two hosts, right? We have the Anopheles mosquito, and in the Anopheles mosquito, it undergoes sexual reproduction. Right? So we say that the Anopheles mosquito is the definitive host. Right? In the definitive host, Plasmodium undergoes sexual reproduction. Right? So here we have fusion of gametes, sperm and the egg, and then producing these merozoites, we call, that are genetically diverse. Right? Every single one of these is different from each other, genetic diversity. Life cycle then, and then these merozoites go live in the salivary glands of the mosquito, and when the mosquito bites a human, these guys swim to the liver and then into the bloodstream, where then they go in and infect red blood cells. And within the red blood cells now, it infects, multiplies many times, bursts, and then infects some other red blood cells. So within the human, it undergoes asexual reproduction. Very quick way to gain numbers, right? And humans are what we call the intermediate host. And and this blood stage is when we see the signs and symptoms of malaria. As red blood cells begin to burst, we get very fatigued, a lot of sweating and fevers, very tired, and it takes about 48 hours to undergo the cycle. So actually the way malaria is often felt is that you have one good day, and then you have a bad day where you feel all the symptoms, and then you have a good day, and then you have a bad day. Right? So it just kind of goes around and around and around. The cycle is complete when a mosquito bites an infected human, and then these guys are sucked up. They'll suck in the gametes, where then they can complete a life cycle, undergo fusion of gametes, and then have a genetically diverse pool that then they can go and bite the next human. Right, so now look at the interplay between that Plasmodium uses between two hosts. One host is strictly used for sexual reproduction. One host is strictly used for asexual reproduction. So Plasmodium cannot spread from mosquito to mosquito or human to human. It doesn't quite work like that, right? Because of the fact that it requires both hosts in order to complete life cycle. It has to bounce between humans and mosquitoes. I mentioned earlier how Plasmodium is difficult to treat because of the fact that its sexual reproduction creates this genetically diverse pool that makes it hard to target any specific structures. So right now, what do you think is the best way to prevent malaria infections or to attack it? Eliminate the host, right? Eliminate the insect vector. And so in places where there are malaria infections are very high, they have many controls, regular spraying of insecticides to prevent lack of mosquito breeding. They find stagnant pools of water where mosquito like to breed and make sure that they can dump out any stagnant pools, prevent mosquito from breeding. The Anopheles likes to bite at night, and so they often sleep with mosquito nets over their beds, again, to prevent this whole transmission. Right? So if you can prevent getting bitten by mosquitoes, you can prevent transmission of malaria. Toxoplasma, or toxoplasmosis, is caused by a microbe called Toxoplasma gondii. Right? This is my favorite microbial disease. There's so many interesting things. Toxoplasma lives in the cat as its definitive host. Right? So what happens in the cat? If you said sexual reproduction, that is correct. Right? So Toxoplasma uses the cat for sexual reproduction. Lives inside the cat, and eventually the cat poops out the cysts. Now the cysts can make its way into the intermediate host, and the mammals are intermediate hosts, and basically other mammals, right? Not cats. So mice and rats are probably the most common intermediate hosts. Cows can pick up Toxoplasma as they are grazing in areas that that cats have made doo-doo on, right? And then Toxoplasma will undergo asexual reproduction in these mammals. And then it'll come back to the cat usually when a cat eats a mouse or a rat, right? As shown in that picture over there, number 5. And then the cat will eat the cysts that have resided into the muscles of the mouse, and then completing its life cycle, getting back to the feline. Again, very important, right? Now humans can get Toxoplasma or toxoplasmosis by eating or inhaling cysts, right? So we can eat cysts if let's say we eat an undercooked cow or an undercooked rat, for those of you who eat those kind of things. Right? So if you eat a rat or a cow that have cysts in them, and the cysts haven't been properly destroyed, you can catch toxoplasmosis. Or you can inhale it, and this might be maybe the most common way, by dumping kitty litter. And when you dump kitty litter, the cyst will blow up into the air, and then you can inhale the cyst, right? And it makes its way into the body that way. Now, for the most part, toxoplasmosis is not too harmful in humans. Right? There's been studies that show. Oh, yeah. Let me go back to what happens in toxoplasmosis. So, toxoplasmosis for things like rats are pretty neat because toxoplasma can live in the nerve cells. And it ends up altering behavior of the rat, right? So normally when a rat or mouse sees a cat, the rat runs away, right? But Toxoplasma, because of the fact that it affects the nerve cells, changes the behavior, and it makes them actually, releases their inhibitions, makes them actually more brave, right? So they're less likely to run away from cats. This, of course, is very beneficial to Toxoplasma because Toxoplasma wants to get back into the cat. So then by releasing inhibitions, making it brave, rats are less likely to run away, and then cats are more likely to eat the rat, right? And then thus completing its life cycle. And this is one of those really cool things where single-celled organisms are able to control the behavior of large multicellular organisms, right? So we always feel like that we are in control, but actually in this case, Toxoplasma is in control. All right, so getting back to humans, when humans catch toxoplasmosis, for the most part, we don't really feel too much, quite benign. Right? But there have been studies that have shown that toxoplasma can cause problems in humans. It can cause some behavioral issues, a lot of moodiness. They say it's highly associated with things like road rage and suicide, that kind of stuff. Makes men more aggressive, women a lot more emotional. And so it does affect our behavior to some extent. But there's a group of people that it's very harmful to, and these are pregnant women. And the reason that it's harmful to pregnant women is because pregnant women the toxoplasma can cross the placental barrier into the fetus and actually attack the fetus and often cause miscarriages. So you'll often see signs in a gynecologist's office where they say, if you're pregnant, don't change kitty litter, because of the fact that you could catch toxoplasmosis and thus harming your fetus. Right? But I always say that if there's a pregnant woman changing kitty litter, there's something wrong with the picture. There's someone out there who's not helping this pregnant woman change kitty litter. Because pregnant women have no business changing kitty litter by themselves. Now, before you go out, go back home and throw your cat away, you don't need to really fear toxoplasma from your cat. Right? If you have a domesticated cat, for the most part, they do not carry toxoplasma. Right? And why do you think that is? That's because, for the most part, domesticated cats don't go running around eating rats. Right? Right? Now, if you have one of those wild cats that run around and eat rats all the time, well, maybe you should get your cat checked out. You can go to the vet and you can get your cat checked out for Toxoplasma. You can even, I think you can get a vaccine. I could be wrong. But you can get your cat checked out for Toxoplasma if it's one of those crazy wild cats. But for the most part, domesticated cats, you don't have to fear. Now, humans are what we call, and whoops, I need to change that. Humans are not intermediate. We call these accidental hosts. We'll refer to that again a little bit later. Because humans don't complete the life cycle. For the most part, Toxoplasma doesn't want to get into humans because it can't get back into a feline, right? Unless we get maybe eaten by a tiger or a lion. In that case, it'd be okay. But for the most part, humans kind of end the life cycle of Toxoplasma. It's not where they want to go. All right, so these are two protozoan diseases. And notice some of the stages here now. So when the cat makes doo-doo, this is our cyst stage, right? This cyst stage allows Toxoplasma to survive outside of the host for a period of time while it's waiting to get picked up and move its way into the rat or some other mammal. So the cyst stage allows it to survive outside of its host and just wait until it can get into the next host, right? So in this case, very important part of its life cycle. All right, so that's protozoans. Let's talk about helminths, or our parasitic worms. Right? These were our multicellular animal parasites that we talked about. For the most part, again, helminths are free-living, non-harmful. But we only care about the harmful ones, right? So we'll talk about parasitic helminths. And parasitic helminths are quite different from free-living helminths. Parasitic helminths often have a very poor digestive system, very poor nervous system. They don't really need to sense and respond to the environment very much. Many of them are not very motile because of the fact that they're simply dependent on getting into the humans. And once they're in the right tissue where they need to be, they don't need to do much except just eat away. But what they do have a very complex reproductive system. And again, just like the protozoans, they'll have be able to perform both sexual and asexual reproduction and involve multiple hosts. Now, helminths can either be monoecious, which means hermaphroditic, or dioecious, which means that we have separate male and female organisms. So we are dioecious. But many parasitic helminths are monoecious, in the sense that they don't need to wait for a mate to be able to reproduce. They have reproductive organisms, both reproductive organisms, and just one. And it makes it a lot easier for it to reproduce. The basic life cycle of helminths are egg, larvae, which are babies, and then adult. And then this adult then lays eggs, and then those eggs grow into larvae and become adults. Two groups of helminths, platyhelminths and nematodes. And the way I like to remember platyhelminths and nematodes is that plat sounds like flat, right? And platyhelminths are the flatworms, or the flukes and tapeworms. Nematodes, the O is round, right? So nematodes are the roundworms. And there are basically two ways that we can catch helminthic infections. Either eating of eggs or cysts, which are excreted in the feces, or eating larvae from undercooked meat. And it's rare to eat the adults, but I guess you could eat the adults as well. But the adults are pretty large, so for the most part, you would see them before you eat them. And just like the protozoans, we are going to look at a couple of three diseases, I believe. This one's called the lung fluke, Paragonymous westermanni. It's a lung fluke, so this is a platyhelminth, flatworm. It lives in humans, and humans are definitive hosts, right? So what happens in the human? That's right, uses a human for sexual reproduction. And so in the human now, undergoes sexual reproduction, and then it's the eggs are released in or excreted in the feces. Right? So as it's growing inside your lungs and causing the problems, it now excretes the eggs, which then we excrete in the feces. And then it uses the mollusks, which now is excreting feces, uses mollusks as intermediate hosts. Oops, why did I go too far? Uses mollusks as intermediate hosts, right? So now here it grows within the mollusk and does its asexual reproduction. And then the cycle is completed when humans eat mollusks, things like crayfish, things like snails, and this is often eating the larvae or the eggs that are within the crayfish or the snail, and then thus completing the life cycle. Right? So, and often this happens when you are undercooking things like your crayfish or your water snails for food. Life cycle is completed, gets back into the human. Maybe a more common disease or common helminthic infection is trichinosis. And this is caused by nematode, right, or roundworm. Trichinosis is caused by Trichinella spiralis, and trichinosis lives in pigs. And in pigs, it produces these cysts that live in the muscles of pigs, and then other animals can get infected by eating pig meat. And of course, humans get infected by eating undercooked pork that contain those cysts. Right? And then it grows inside your intestines and then causes some major problems. Right? So trichinosis is very common when you're eating raw or undercooked pork. Now, trichinosis or Trichinella is very sensitive to freezing. Right? So most of our pork that we eat has actually been frozen first. And this helps kill off the trichinosis, right? The Trichinella. Either that or the pork has to be screened very carefully, right? So trichinosis is more common when you eat pork that's wild, right? So for feral pigs, for example, if that's what you're into eating, or freshly slaughtered pigs, in that case, you got to make sure that you cook your pork properly. Do not eat it rare; should always be well done. Our last helminthic infection we're going to talk about are rat lungworm, or caused by Angiostrongylus. Now, why are we talking about a disease that affects rats? Well, they're somewhat important to humans. So for one last time, rats here are the definitive hosts, right? So what happens in rats? Yeah, that's right, sexual reproduction. And so it grows in the rats, and then the rats, they make doo-doo, and then excrete the Angiostrongylus in the feces, where it lives in snails. Snails now are our intermediate hosts, right? And so, what happens in snails? Asexual reproduction. And here now, the larvae are excreted in the snail feces, and then these snail feces, and then when rats go and play around in whatever the snails are playing around in, it gets back into the rat, and thus completing its life cycle. Now, humans can get Angiostrongylus, or rat lungworm, from eating larvae from raw or undercooked snails, if that's the kind of thing you're into eating, or the snail has contaminated water or vegetables. And in this case now, humans are a true accidental or incidental host because they are really not part of the life cycle. Right? At no time does Angiostrongylus ever want to be in humans because it does not complete its life cycle. Right? But if you, as a human, get Angiostrongylus, all of a sudden now you can get sick, and it causes some major issues. It goes to your brain, Angiostrongylus does, and causes things like puts you into comas. And I heard it's really, really tough disease. And why are we talking about rat lungworm or disease that causes problems in rats? Well, it's kind of got a local flavor to it, right? So there's 3 cases in the Big Island in 2008, 3 more in 2011, and a Minnesota man in 2012. All contracted rat lungworm. This Minnesota man, he had trouble. He had to learn how to walk again, right? Because it really messed up his brain. And all, 7 cases here have been traced to homegrown vegetables, right? And this Minnesota guy, he came to Hawaii to learn about organic farming, and he ended up contracting rat lungworm disease. So, what's the lesson here with everything we've talked about in this chapter? Make sure you wash your vegetables properly. Just because you grow it in your garden doesn't mean it's clean, right? There's things going around in there, so just wash them. Doesn't require a soap wash. Right? Just rinse them out properly, and make sure you cook your food properly. Cooking food, washing vegetables is the way to make sure that we get rid of a lot of these things that are living on our foods.